Welcome, everybody. It's my pleasure to introduce Dr. Andrew Routh. Dr. Routh obtained his undergraduate degrees in cellular and molecular biochemistry from the University of Oxford, Lady Margaret Hall in the UK. Subsequently, he moved to the Medical Research Council Laboratory of Molecular Biology, University of Cambridge King's College, obtaining his PhD under the supervision of Dr. Daniela Rhodes. Here, he studied the impact and roles of linker histones, nucleosome repeat length, and histone post-transcriptional modifications upon the compaction and structure of the 30 nanometer chromatin fiber. Dr. Routh then moved to the Scripps Research Institute, now Scripps Research, as an EMBO postdoctoral fellow to study RNA virus replication, recombination, and virion assembly under the mentorships of Professor Jack Johnson and Professor Bruce Torbay. Dr. Routh took up his current position as assistant professor at UTMB in the Department of Biochemistry and Molecular Biology in 2015. In his lab, they combine molecular and cellular virology, next-gen sequencing, and computational biology to study model RNA viruses, such as the flockhouse virus, as well as human pathogens, including HIV, coronaviruses, and arboviruses. Together, these provide clinically important and experimentally tractable model systems for the exploration of the principles of RNA virus biology and evolution. His latest work has been focused on applying his ClickSeq-based methods for virus genomics to study RNA recombination and defective RNAs of SARS-CoV-2 in the current pandemic. In addition to his biotech startup based in Galveston in addition to his academic lab, Andrew, together with Dr. Elizabeth Jaworski, founded ClickSeq Technologies, a biotech startup based in Galveston that uses the ClickSeq platform to provide next-gen sequencing services and bioinformatics analysis. And before we begin, I just want to remind everybody that you can put your comments or questions in the chat, or you can wait until the end and unmute yourself to ask your questions. So now I'll go ahead and hand it over to Dr. Routh.
Thank you very much for the introduction. It's a real honor to be invited and present my work to you. So today I'm going to talk about some of our coronavirus sequencing work that we've been doing here on the island of Galveston. And as you well know, coronaviruses, especially SARS-2 coronavirus, don't need a lot of introduction. But one of the things I'd like to point out is RNA viruses are really remarkable in their ability to evolve. We sometimes call them the masters of evolution, and SARS-CoV-2 is no exception to that. And what's incredible about what's happening during the pandemic is just how much sequencing information we have on SARS-2 coronavirus. So just this morning, I looked at the GISAID database for how many coronavirus genomes there were, and there's almost 5 million deposited sequences, complete genome sequences deposited in the database. So extremely rich source of information on how RNA viruses are evolving. And generally speaking, when we're thinking of RNA viruses evolving and what you and I might immediately think of when we think about SARS-CoV-2 evolving, we think about variants of concern. We think about things like the alpha variant or the delta variants that have been sweeping across the globe at the moment. And in each of these, so for alpha, for example, and delta, they have characteristic mutations in the spike proteins. And it's often been these characteristic mutations that we're generally focused on, we tend to be most concerned about, because those might be the ones that are the breakouts from vaccines or from our own immune system. And so they might represent the next wave of the next virus. And so we're all very acutely aware of how important mutations are, especially in the spike protein. There's a whole other branch of RNA virus evolution and mutations that is really important that I'd like to talk about today. And that's really about RNA recombination. And RNA recombination, quite simply put, is just the recombination between two different genomes to create a new genome. And so this is an incredibly important mechanism in the evolution of RNA viruses. And so in the most basic sense, we have what's called homologous recombination. And so if you have two RNA templates in a single cell, it's possible during the replication of those templates for the polymerase to jump from one template to another one in an event called a template switching event. And what that can do is that can actually shuffle mutations between genomes. So you can get these reshuffling events, which sort of either enrich for useful or advantaged mutations, or it can actually remove deleterious mutations. And so whilst the actual event of recombination itself, there isn't any evidence of it in the resulting virus sequence, the entire virus is actually a new chimeric virus. And it turns out this is incredibly frequent. So when you do experiments to measure these rates in cell culture, for example, with coronaviruses, you can show that every single coronavirus RNA genome that arose, about 25% of those are a product of a recombination event. And so it's an incredibly frequent, incredibly important mechanism in the evolution of RNA viruses. And so homologous recombination is when recombination occurs between homologous sites, so the same site on two different genomes. But another important aspect of recombination is what's called non-homologous recombination. So as you imagine, that's the non-homologous site. So if you have a recombination event between two different parts within the viral genome, you can get these quite complex rearrangements, including deletions or insertions in the viral genome. And so those non-homologous recombination events can generate novel genomes might be missing parts of the original virus. So you have these deletions or insertions in the virus genome. And they can also give rise to this phenomenon of defective virus genomes, which I'll mention in a moment. And so a really important aspect of recombination is what happens when you have two different viruses getting into the same cell. So if you have a co-infection with two different viruses, you can still have recombination, but between the two genomes of those two different viruses. And that essentially generates a new chimeric virus. And there are lots of examples of this in nature. So rhinoviruses is classic. There are hundreds of strains of different rhinoviruses. And we can very clearly see by looking at the sequence information from those viruses that some of them are the products of recombination from two different viruses. So recombination is important for multiple levels in terms of adaptation by deleting and accumulating these advantage mutations, in terms of structural rearrangements, and in terms of co-infections. And coronaviruses do this a lot. So in the origin of SARS-2 coronavirus, one of the original postulates of where the virus came from was during a co-infection of two different coronaviruses presumably in a bat. And so it was noticed that part of the spike protein of SARS coronavirus, the majority of it had all a sequence homology to a bat coronavirus, but there was a large portion of the receptor binding domain that had a sequence homology to this pangolin coronavirus. And so it speculated that perhaps the SARS-2 coronavirus was actually a product of recombination between two other coronaviruses in a host, some hosts, such as possibly a bat. But we also know that recombination has taken place in the origin of the pandemic, because in the spike protein of SARS-2 coronavirus, there's this curious insertion in the spike protein near the furin cleavage site. And so this infamous insertion, this PRRA, looks like it was essentially a duplication of part of the viral genome. But this duplication, this insertion, is really another recombination event. And so there's multiple lines of evidence to suggest that these recombination events are really what are giving rise to these novel variants and giving rise to potentially zoonotic viruses like SARS-CoV-2 in the first place. And so we've got this is a very fundamental concept, very fundamental principle of how RNA viruses arise. It's important to understand the molecular mechanisms that drive these processes. And so RNA recombination is very common during just the normal cell cycle, normal replication cycle of the virus. And so I mentioned a moment ago how we have homologous recombination and we have non-homologous recombination. And what's really interesting is non-homologous recombination can give rise to these virus genomes that are essentially missing portions of the original virus. So they're defective in some way. So a recombination event will delete a part, or it can insert a part of either the host or a virus RNA into the virus genome, generating these defective RNAs. And these defective RNAs have been noticed in many, many different virus systems. So we see them in poliovirus, where there's parts of the structural genes have been deleted. We observe them in the flockhouse virus, where multiple parts of the virus gene have been deleted. And then there's many other examples, like in influenza, where you essentially get recombination events stitching together the end and the beginning of some of the RNA segments of the virus genome. And what's really curious about these defective RNAs is that whilst they spontaneously arise due to recombination, they can actually get maintained during passaging of the virus. So these defective genomes retain the important parts of the genome that are required for the replication of that genome, as well as encapsulation into the virus particles themselves. So these defective RNAs, these defective genomes, pretty much parasitize the wild-type virus. They can actually piggyback along with the wild-type virus and act as a passenger to the infection. And so these defective RNAs are extremely interesting. They're teaching us about where recombination events occur. They teach us about the frequency of recombination events. So they're really useful tools for studying RNA recombination. But in the last few years or so, it's also emerged that defective RNAs, which are products of recombination, actually can change the outcomes of disease as well. And so a really nice example of this is, again, looking at influenza virus, another respiratory pathogen. So during the 2009 H1N1 pandemic, swine flu pandemic, it was noticed that the wild type strain of the flu virus would readily generate defective RNAs of one of the segments of the viral genome. And so when they put this in cell culture and they showed that this virus generated lots of defective genomes, these defective genomes are actually immunostimulatory. So it was hypothesized that these novel defective genomes had some kind of RNA secondary structure that stimulated the immune system in the host cells, and that's actually helped with virus clearance. And so in that state, there was a heightened immune response, and in an animal model, there was actually better survival of mice in an animal model, and in humans, it was associated with milder symptoms. But what was interesting is that if you engineered a point mutation into the polymerase of that particular influenza virus, it actually generated fewer defective RNAs. So these defective RNAs are generated spontaneously during replication. So by altering the polymerase, they reduce the rate at which these defective RNAs were generated. And so there's fewer defective RNAs. That meant the immune system was less well stimulated in those cells. And then in a mouse model, it actually resulted in more animal death. And then in humans, it was associated with more severe outcomes. And so we can see through this example that the frequency of defective RNAs and the frequency of RNA recombination was actually correlated with the outcome of disease. So again, RNA recombination is not just pivotal for RNA virus evolution, but it's also important to understand why some people get sick and other people get sicker. So coronaviruses do a lot of RNA recombination. As I mentioned, they do a lot of homologous recombination, and this is part of their normal viral life cycle. They do it a lot inside cells. They can generate these variants, these novel variants characterized by insertions or deletions. The original strain of SARS-2 has this novel insertion in the furin cleavage site, but more recently, the variants we've seen, like the alpha variant and the delta variant and so on and so forth, many of those also have characteristic deletions in them as well that have been generated through RNA recombination. And then coronaviruses also generate a lot of defective RNAs. So just like the case with flu, we know from other coronaviruses that have been passaged in the lab or sequenced in the lab that they form these defective genomes, the missing parts of the defective RNAs are missing parts of their genomes. And so these defective RNAs in coronaviruses have been demonstrated to interfere and attenuate the wild type virus. So we know that coronaviruses generate defective RNAs. Furthermore, in coronaviruses, they have this really interesting facet of their life cycle where they have a number of protein open reading frames sort of hidden in the 3' end of their genome. And in order to express those proteins, they generate what's called a subgenomic messenger RNA. And these subgenomic messenger RNAs are essentially recombination events. It's kind of like messenger RNA splicing in human hosts, but in the viruses, it's not splicing, it's a recombination event. And it stitches together the first 70 or so nucleotides of the 5' UTR right to the beginning of those open reading frames. And this gives rise to each of these smaller transcripts, which each encode for important proteins. For example, the spike protein and nucleic acid. And so in all these cases, recombination is critical for the virus life cycle as well as for its evolution. And so we wanted to ask some basic questions to study RNA recombination in coronaviruses. We want to know what's the role of RNA recombination in pathogenesis. We want to know, does recombination play a role in the emergence of new strains of coronavirus? And then if it does, we want to know what are the molecular determinants of that. So what is it about coronavirus and the way it replicates that it selects certain sites for recombination? Or what's the frequency of recombination in different strains? And so one of the main tools we use to study this is with next generation sequencing. And next generation sequencing is a very, very powerful method, but it's also very quantitative. So the useful thing about next generation sequencing is if you take a virus, a sample, and it could be pretty much anything. So in this case, for example, we've taken some purified virus. You can take that purified viral RNA and you put it into your pipeline. You fragment that viral RNA into lots of small little chunks, and you append sequencing adapters onto the ends of each of those little fragments of RNA. And then you reverse transcribe them and generate a library. And so your library is essentially representative of all of the fragments of RNA from the original sample, but with your sequencing adapters on either side. And that allows you to sequence in a very high throughput manner and in a quantitative manner your virus. And using bioinformatic protocols, we can align those small fragments to our viral genome in order to reconstruct our original viral genome computationally. And now, because it's quantitative, and because it's what you put in is essentially what you see, so what RNA you put into your system is essentially what you end up sequencing, you can do some very interesting things. And you can measure the frequencies of recombination events. You can measure the rates of point mutations, such as nucleotide mutations that arise throughout the genome. And you can do it in a pretty quantitative manner. And so previously in our lab, we've worked quite hard to generate bioinformatic protocols to do this. So one of the challenges of sequencing recombination events in RNA viruses is just how unpredictable they are. So they tend not to occur at very specific sites. They tend to occur stochastically and at many different points throughout the genome. So when we look at programs that map, for example, splicing events in human messenger RNAs, those programs that map those sequencing data, they generally follow rules such as they know where the splice donor site is going to be and the splice acceptor site is going to be. And we always know that the acceptor site is going to be downstream from the donor site. With recombination RNA viruses, that's really not the case. You could see insertions or deletions, and they could occur at different sizes at different sites. And so we really needed an agnostic tool that could map all of these unusual recombination events. And so to do that, we used some Python scripting, and we generated this software package that we called Virema for Virus Recombination Mapper. And the principle is quite simple. So if we take a sequencing read, it essentially uses Bowtie, it bootstraps this program called Bowtie, to map that sequence read to the virus genome. Now, if it finds a part of that read that doesn't map to the virus genome, all it simply does is extracts that unmapped region and then tries to remap it in a separate round of sequence alignment. And so it's pretty straightforward, really. But the important point is that second mapping, when you map that second part of that read, is completely independent of the first mapping. And so you could find any kind of recombination event, whether it's upstream or downstream, even if it's between different viruses or between the host and the virus. And so this tool gives us the ability to map recombination events regardless of their identity or what sequence happens to be there. And it's completely agnostic to the type of recombination event or splicing event that you would have expected. And so Stefania Sotchev is a graduate student in the lab. She generated some essentially simulated data, some synthetic sequencing data, and used that to demonstrate that this pipeline really accurately captures recombination events in coronavirus genomes. And so we can use it to map around 95% efficiency sequence reads in our data sets to recombination events in the coronavirus genome. So for the last few years, we've been collaborating with Mark Dennison and Jennifer Gribble at Vanderbilt University. And they, for a long time, have been studying RNA recombination and evolution in murine hepatitis virus. And so murine hepatitis virus is another coronavirus, and it's a very important model system for studying coronaviruses and their evolution. And so one of the things that they've been doing is passaging MHV for several, I think, months, maybe even years in cell culture over time. And one of the things they've been interested in is actually characterizing the rates of RNA recombination in those cells. And so we got together a few years ago, and we're using our bioinformatic tools to measure RNA recombination rates in murine hepatitis virus. And so what Jennifer did is a pretty simple experiment really to start with. She took cells in culture in a monolayer and infected them with murine hepatitis virus. And after about 3 days of infection, she extracted the supernatant materials and then sequenced everything that was in that supernatant. And we did a combination of nanopore sequencing, so long read sequencing, as well as Illumina sequencing for short reads. And with these short reads, we can use those to map RNA recombination events using our Virema program. And so what we found was quite interesting. So by taking all of these recombination events and mapping to the virus genome, we found many, many recombination events with quite interesting distributions in where they were found. And so we found around 250 recombination events for every 10,000 reads that we had in total. And we had around 30 million reads in total. So we found thousands of recombination events. Now, this data is very complex, and there's lots of start sites and lots of stop sites in recombination events. So it's quite difficult to that's what I'm looking for, like illustrate the data. But one of the ways that we do it is using these heat maps. And so if you bear with me for 1 second, I'll explain how we plot these data. And so on the right here, I show what is essentially an example of a recombination event. You've got your virus genome, and then on the top, you've got your sequencing read, where we've mapped a breakage in that sequencing read corresponding to a recombination event. So on the left-hand side, this is our donor site. This is our 5' site, or our start site of our recombination event. And on the right-hand side, we have the stop site. So our recombination event goes from this 5' site to the 3' site. And then we have thousands and thousands and thousands of these reads. And so we have multiple different start sites and multiple different stop sites. And so we can plot that on a scatter plot like this. So on our x-axis, we plot the stop position, so this site here. On the y-axis, we plot the start position. So in other words, let's say you had a recombination event that deleted nucleotide, say, 15,000 to nucleotide 20,000. Well, then you plot that on your plot with your stop site being 20,000, which would be here, and your start site being 15,000, which would be around here. And then you know that you had a deletion at that site. So each of these points in our heat map corresponds to a novel recombination event that we found. And what you can quite clearly see is although there's thousands of recombination events, they're not randomly distributed. There really are hotspots across the genome. And so quite interestingly, the majority of recombination events, the stop sites are actually found in the 3' UTR. So most of the recombination is going from the virus genome and jumping into the end of the virus genome at the 3' UTR. And we see this massive enrichment for recombination events at these sites. We also see a lot of recombination events from the beginning of the genome, so the 5' UTR. And this includes those subgenomic messenger RNAs I showed at the beginning. And so importantly, this is telling us that RNA recombination isn't random. It's really favored to specific spots. Not only that, but with our data sets, we can measure the rates of RNA recombination, in this case being around 250 reads, recombinant reads per 10,000 mapped wild type reads. And so in coronaviruses, what's unusual about coronaviruses is the way that it replicates this genome. So coronaviruses are unique in that the polymerase, the replicase, actually has the ability to proofread itself as it's replicating. So this is completely unique to RNA viruses and might be the reason why coronaviruses are able to maintain such a long genome. And so they have this protein called NSP14. It's an exonuclease. It's thought to be associated with the polymerase during replication. And it's thought to sense mismatching in the genome as it's being replicated. And if it finds a mismatch, it uses its endonuclease to remove that mismatched nucleotide, and then replication can start again. And so this proofreading capacity is a really important property of coronaviruses that allow it to have such a low error rate compared to other RNA viruses. And so we tried a simple experiment. We assumed, well, if you knock out the proofreading enzyme for the polymerase, well, then you'd get more error. And if you get more error, you see more recombination in your virus. But what was surprising is when Jennifer did this experiment, she knocked out the XON. So she introduced a mutation that made it catalytically null NSP14. In fact, the recombination rate was dramatically reduced in these viruses. So the wild type was around 250 recombination events per 10,000 map reads. With the mutation, it was lower than 50. So it's about a 5-fold reduction in the rate of recombination just by 1 point mutation in the proofreading polymerase. And then we look at these heat maps again. Here's the wild type. And then on the right, you see the mutant. You can see the distribution of these recombination events is pretty similar. So we still see this enrichment for recombination events at the 3' UTR. And so although the rate is much lower, the sites at which they occur is pretty much maintained. And so this is pretty intriguing. This is really telling us that there are point mutations that are variants essentially in the polymerase which can control the rates of RNA recombination for a virus. And in the case of MHV and this proofreading enzyme, it's really curious that the enzyme that is normally responsible for reducing the error, is normally responsible for controlling the error rate. When we knocked it out, in fact, we saw a lowered RNA recombination rate. So sort of opposite to what we might have expected. And once we completed this work, this is around at the beginning of 2020. So it's just as the coronavirus pandemic was starting. And so we immediately turned our attention to other coronaviruses. And so we repeated these experiments. We essentially did the same thing. We took cells and culture, and then we infected them with either Middle Eastern respiratory virus, so that's MERS, or with SARS-2 coronavirus, which we had from an isolate from the Washington strain very early on. And we infected cells and culture, and we did the same experiment as we did with MHV. We extracted the supernatant after a couple of days of infection, and then we deep sequenced everything in the supernatant. And then we used our bioinformatic pipelines to map RNA recombination events in the genome and to measure the frequency of RNA recombination. And again, what we found was very striking. So in MERS coronavirus, we found the recombination rate was actually quite low. So it's similar to MHV. It was around 50 recombination events per 10,000. And the distribution of recombination events is also quite similar. So we see this enrichment of recombination at the 3' UTR with MERS coronavirus. But what was striking with SARS-2 coronavirus, the rate was 10 times higher than we saw with MERS. And so these are in the same cell type. We see this much higher recombination event with SARS-2 coronavirus. Again, the distribution was quite similar. So we see this enrichment for recombination events at the 3' UTR. And so we can look into this data a little bit more carefully and say, well, we know there's a bias to the 3' UTR, but is there any other information that we can glean from these reads? Is there any nucleotide specificity? And because we've got two very similar viruses, we can compare them and ask, is there anything specific or novel about SARS-2 coronavirus that is giving rise to this increased RNA recombination rate? And so what we did is we looked at the sites of all of the recombination events. And so remember, we've got thousands, tens of thousands of unique recombination events that we're measuring here. We can look at the nucleotides that are found either upstream of the recombination event or downstream of the recombination event, and we can plot their frequencies. And so in black here, this is the distribution of either A's or C's or G's or U's upstream of the recombination events. And then in purple is the traces that we saw with SARS-2 coronavirus. And what was really striking with SARS-2 coronavirus, we see this massive spike, this enrichment of uracils upstream of our donor sites for our recombination events. And so if you have an enrichment of U's, of course, you've got a concomitant depletion in G's and C's and A's as well. And this was unique to SARS-2 coronavirus, which is intriguing. And then not only is this found upstream of the recombination event, we also found the exact same feature downstream of the recombination event as well, implying this microhomology at that recombination site. So to sort of summarize, from cell culture alone, we found that the RNA recombination rate of SARS-2 coronavirus is much higher than that of other coronaviruses, including MERS and MHV. Not only that, but it really appears to be specific for U-rich tracks. So for some reason, we're seeing an enrichment of RNA recombination rates at U-rich tracks in the SARS-2 coronavirus genome. And so this led us to ask, well, maybe this is an artifact of the cell culture that we're working with. These are cells in culture, we're passaging for a few days, we're amplifying the virus. There might be something unusual about cell culture that's enriching for these unusual recombination events. And so like a lot of people during the pandemic, we turned our attention to sequencing clinical isolates of SARS-2 coronavirus. And this is important for two reasons. One, to measure rates of RNA recombination in clinical cases in human hosts, but also to characterize novel variants as they're arising, many of which, as I mentioned before, were the product of RNA recombination, had recombination events in them. I'm getting a few questions in the chat, but I think I'll come to them at the end because I'm going to address some of these questions as I'm going through, and then I'll get to them. Okay, so sequencing coronaviruses in patient samples. So the majority of sequencing that has been done on SARS-2 coronavirus, like I mentioned, the pandemic is an extraordinarily unusual situation, just how much sequencing has occurred in such a short period of time. SARS-2 really is one of the most sequenced viruses that has ever existed. And one of the reasons for this is probably because of this protocol called the Arctic protocol. And so it's an extremely powerful, very robust method for sequencing viruses. And the basic methodology is to take your input RNA and then reverse transcribe it with random primers. And then the Arctic protocol has designed these pairs of primers that recognize the virus genome. And so these pairs of primers, you amplify in a PCR reaction to generate lots of small amplicons, around 200 to 600 nucleotides in length. Now, what's important is these pairs of primers have been very carefully designed to generate amplicons of specific sizes so that once you sequence all of these amplicons, they actually overlap. And it's from these overlapping amplicons you can reconstruct the original virus genome. And you can do this either on a Illumina platform with short reads, or you can actually adjust the primer sets that you use. You can redesign your primers so they're further apart and generate longer reads, and then you can sequence on the nanopore pipeline as well to get longer reads. And both of these methods have been used extensively. Like I said, there's been around almost 500 million genomes assembled so far in the pandemic. But as powerful as these approaches are, there's still a few limitations. So one of those being that by having to design pairs of primers, that puts lots of limitations on where those primers can occur. You don't want your primers to be overlapping with highly variable regions or where you might have variants of interest arising, mutations of interest arising. It also constrains what sort of recombination events you might be able to find. And so in the case of subgenomic messenger RNAs, one of the most common recombination events in coronavirus, you can design a primer for your 5' UTR to help you capture most of those recombination events. But if there's anything unexpected, if there are unusual RNA recombination events, unless your primers happen to be upstream and downstream of that recombination event, then you're going to miss it using the Arctic protocol. So we wanted to address this. We wanted to be able to capture all the recombination events in clinical samples. And this is challenging. You have a tiny amount of material in a clinical sample. It's often highly degraded. It's often coming from an NP swab, so it's got all sorts of mucus and other proteins in there. The RNA is very poor quality. It's really hard to design robust methods to amplify the virus genome. So you can't just go in and sequence everything. You've got to use these template-specific primers to amplify the virus that you're interested in. And so the way that we did this is with a method that we call ClickSeq. And so to generally introduce how ClickSeq works, is a slightly different way of making next generation sequencing libraries. And we've used it a lot in our lab to study RNA viruses. And so very briefly, ClickSeq works by, without any fragmentation, we do these reverse transcription reactions using random primers. But during the reverse transcription, in addition to having deoxynucleotides, we also spike in these azidonucleotides as well, so these AZNTPs. And these azidonucleotides are a certain ratio, around 1 to 20 of azido to deoxynucleotides. What that means is that these azidonucleotides, which are terminating nucleotides because they have this modification, they'll get stochastically, they'll get randomly incorporated into the cDNA as you're replicating the template. So your cDNA will be terminated by this azidonucleotide, so you'll end up with this azido group at the end of your cDNA fragment. Now, because the priming is random and the termination is random, essentially what that gives you is lots of random fragments of the original RNA that are in cDNA form, and with that azido group at the end. Now, this azido chemical group is very useful because we can exploit click chemistry to ligate on our sequencing adapter. And so we buy these sequencing adapters from IDT, and they have this hexanol group on the end. So that's a carbon triple bond. And this carbon triple bond can very specifically react with that azido group in the presence of copper. And so we just, quite simply, we take our azido-terminated cDNAs, we add our alkyne modified sequencing primer, we add copper, and that reacts the two together, and we end up with a piece of single-stranded cDNA, which now has sequencing adapters either side of it. And so this removes all of the enzymes that you might use for ligation, and also removes any kind of fragmentation that you might use in your viral genome. And so we've shown through multiple other experiments that this also prevents artifactual recombination in your data. By removing this fragmentation, by removing ligases, you really reduce the rate of artifactual recombination in next generation sequencing, which can be pretty problematic. And so this is a really nice platform for sequencing. And like I said, we use it for RNA viruses, but the principle is very flexible as well. So we can pretty much target anything we want to by redesigning the primers that we use. So if we use random primers, obviously we target everything randomly. But we also develop methods to target the poly A tail using what we call poly A ClickSeq. And so this is also a robust method that we use for sequencing messenger RNAs. So around the beginning of the pandemic, essentially we just adjusted these protocols, and instead of having primers that target the poly A tail, we designed hundreds of primers that target the virus genome instead. And so what we end up with is a scheme like this. If you have your virus genome at the top, we have hundreds of these primers scattered across the virus genome. We do a reverse transcription just like in ClickSeq. And so we generate again a cDNA amplicon, just like the Arctic protocol. But instead of having pairs of primers, now we only have one primer per amplicon. And the other end is generated by that stochastic incorporation of that azidonucleotide. Now, this is really important because what that means is that azidonucleotide can be incorporated anywhere. It doesn't matter if there's a recombination event there or some kind of nucleotide variant or something unusual, it'll get incorporated regardless, as long as your primer can bind to that template. And there's a few other qualitative improvements to the protocol as well. So we can also control for some PCR errors as well by having unique molecular identifiers in our click adapter. And so we validated this very quickly, very early on. So we got some isolates from the receiver, the World Reference Center here at UTMB. And we showed that when we sequenced on the MiSeq, we got quite nice data. So here in orange is depicted our read coverage across the coronavirus genome when we're sequencing with the Illumina platform. And what we see is this sawtooth-like pattern, what we call a sawtooth pattern. And what this is sequence data upstream of each of our primers that we designed in our tiled ClickSeq approach. And so then with nanopore sequencing, you can see in blue, because the reads are longer, we get a slightly smoother profile. So we lose those saw teeth, and we get a much smoother profile across the entire genome. Now, importantly, when we zoom in, what this shows us is that each of these saw teeth are actually overlapping each other. So here I color in blue all of the sequence data that arises from primer 1, and in orange, this is all the sequence data that arises from primer 2, and green from 3, and so on and so forth. And so each primer generates a chunk of sequencing data, which is overlapped from the sequencing data from the previous primer. So just like the Arctic protocol, we get overlapping context, overlapping sequencing data that we can use to reconstruct the viral genome. And so since this time, we spent a lot of time optimizing these primers. In our current scheme, we have around 335 unique SARS coronavirus 2 primers that are strewn across the virus genome. And so we have what we call a version 3 tiled click-seq approach for sequencing coronaviruses. And that's shown here in this pink. And you can see that's actually quite nice smooth coverage over the virus genome. We validated this quite extensively. So using multiple isolates of viruses obtained from the receiver center here, we sequenced them both with tiled nanopore sequencing, random prime sequencing. We've done it at Illumina. We've also done it with the Arctic protocols. And we've shown in every case, we retain the same virus genome. So we get the same correct virus genome from each method. And so now that we have this method, we can start to sequence clinical isolates. We can go into our clinical populations and start sequencing coronaviruses to ask the question about how RNA recombination changes amongst clinical samples. And so in collaborations with Dr. Ping Ren and Dr. Scott Weaver here at UTMB, we were contracted by the CDC to do this work. And so since around, I guess, June or May, we've we've had access to thousands of nasal swabs from patients who were for routine diagnostics. And so we're able to get those samples and take the MP swabs from those patients, extract the RNA, and do the sequencing. And so one of the first things we do is just a reverse transcription just of the spike protein. So we do a preliminary sequence alignment just of the spike protein using Sanger sequencing. And so this is work done in Scott Weaver's lab. And from there, we can determine what variant essentially we have. And they've been doing this very routinely throughout the pandemic in Galveston for the last few months. And so we can trace the frequency of the different variants as they're arising over time. And just like everywhere else in the US, we saw this big burst of alpha variant, or UK variant, around early April, May of earlier this year. And then around July and August is when Delta started to sweep through and overtake pretty much everything else that we've seen. And so Delta is pretty much the only thing that we're sequencing now anymore. And I think the last sample that was non-Delta was back in August that we sequenced, here in Galveston at least. And so this is just with Sanger sequencing for variance determination. So we can take all of these samples and we can do our tiled click-seq approaches to sequence the whole genome and measure rates of RNA recombination. And so we validated this. And with samples with low CT values, so anything below a CT value of around 25, we get very good genome coverage. We can get about 100% genome coverage. Once we get over a CT value of 25, we begin to lose some of the sequencing coverage, and it gets much harder to do. And with this, we can reconstruct our viral genomes. And so, like many others, we've been using the NextStrain platform for doing phylogenetics. We can reconstruct our viral genomes and just simply ask, what variants did we have? And like I said, the majority of our recent samples have been with Delta, but we also sequenced a lot of Alpha. But importantly, of all the samples that we sequenced, we're seeing a different distribution of different variants across them. So out of all the samples, we've got this quite rich database of tiled click-seq sequence samples from a range of different variants. And so this allows us to go in and ask about rates of recombination. But there's one other important feature with the tiled click-seq is that we can sequence all the way up to the 5' UTR of the virus. It's not just the whole genome. We can sequence, well, it really is the whole genome. We can sequence the entire virus plus the ends of the genome. And what we found with tiled click-seq is that we could sequence the entire 5' UTR of these alpha variants, these UK variants. And what we found is in every single case that we sequenced, the second nucleotide was mutated away from the reference. Now, this is quite important because with the Arctic approaches, you have this primer which is designed for the 5' UTR. So the 5' UTR is often missed when using the Arctic approaches. But with this tiled click-seq, we can find these mutations right at the beginning of the 5' UTR. And every single alpha variant we sequenced had this mutation. Furthermore, more recently, we've done a lot more delta sequencing. The same is true for delta. Every single delta variant had this mutation, this U to C mutation at the second nucleoside of the genome. And this is important because the 5' UTR has many functional elements that are important for regulating transcription and translation of the genome. And so it's curious, we see the same mutation both in alpha and in delta. And if we go back to our phylogenetic tree, you can see that delta and alpha really are on different lineages. So it appears like the same mutation arose independently. And essentially what was the third and fourth wave of the virus that we saw in the US, at least. So we don't know what the biological function of this mutation is, but we're looking into that. Now, with these hundreds of strains that we sequence here at UTMB, a number of them are actually from vaccinated patients as well. And so I'm indicating those here with these little stars. And so we can see each of these samples here were from vaccinated patients. And early on, after the vaccine rollout, we're very concerned that the spike protein might start evolving more quickly in the presence of a vaccinated population. We might start to see novel variants of the coronavirus break out as an adaptation against the immunological background of the virus. And so it's very important that we start sequencing these samples from vaccinated patients to try and observe what was arising. Thankfully, so we've only sequenced a handful of samples from vaccinated patients. And importantly, in very few of them have we really seen anything unusual. So oftentimes the sequence that we generate from this vaccinated patient is very similar to those we find from non-vaccinated patients. But there's a couple of notable exceptions to that. And one is this delta sample here that I've starred. And so if we zoom in on this region here, I just want to look at the samples that we have in this region here. So here's a collection of patients that we sequenced coronavirus from. And this one in the middle was from a vaccinated patient, and the other three were not. They were from just normal unvaccinated patients. And what was interesting about this sample from a vaccinated patient, we found out it had two unique mutations. And so one was a spike mutation, but it was actually at nucleotide 1165. So it's right at the C terminus of spike protein. And so this is what the structure of spike looks like. So that mutation is actually outside of the resolved structure of the protein. It's probably in this transmembrane domain that anchors spike into the membrane. And so it's interesting that this vaccinated patient would have a mutation there. But the additional thing that we found was actually a recombination event. So we saw this massive deletion in ORF7 of the coronavirus genome. And so this patient had an 82 nucleotide deletion that removed pretty much the whole of the ORF7 protein. Now, ORF7A is an accessory protein. It's not required for replication of the virus, but it's still interesting that this virus would be viable in a patient in spite of the fact that it's missing part of its genome. And it's also curious that we found it in a vaccinated patient. So of course, we're following up on this. We're generating these mutants and passing to them, characterizing them in cell culture to see if this removed ORF had any change in the pathogenesis or the virulence of the virus itself. But this is a really key demonstration of how recombination can give rise to a novel variant, which might have given rise to a vaccine breakthrough. Okay, so as I mentioned, one of the whole reasons we want to do tile-click-seq in the first place was to find these recombination events. And above in our vaccinated patient, this is one very clear example where we had a recombination event that was penetrant. I was 100% of the virus had that deletion. But we want to be able to detect all the different little recombination events in the background as well, all the really rare ones, all the really unusual ones, and the defective RNAs that might be present. And so tile-click-seq provides a really robust method for characterizing those recombination events. So we went into our clinical samples and we measured the frequency of recombination in all of our clinical samples. And that's, again, what I'm showing here. So these are our heat maps, but instead of from tissue cell culture material, this is now from all our clinical samples all averaged together. So again, it's a heat map. And what you can see from 100 different samples, we saw a very similar distribution of recombination events in clinical samples as we saw in the cell culture material. So first of all, there's this very strong enrichment for recombination events in the 3' UTR. There are sometimes duplications, sometimes deletions, and it shows us the 3' region of this coronavirus genome is highly variable. Similarly, we see this little block here. We see a lot of recombination events between the middle of the genome, around nucleotide 6,000 or so, to the end of the genome. And so these very frequent events that occur in multiple samples, these are probably what's corresponding to defective RNAs, which I mentioned at the beginning might have an outcome, might be able to attenuate the wild-type virus. And then finally, of course, we're detecting all the subgenomic messenger RNAs as well. And there are many non-canonical subgenomic messenger RNAs, as well as the expected messenger RNAs. But because we have multiple clinical samples, because we have a very quantitative sequencing method to measure the frequency of them, we can measure the frequency of each of these subgenome messenger RNAs across all our samples. So for example, in spike here, we can see that the spike protein is probably the most highly expressed subgenomic messenger RNA, and the nucleocapsid is also one of the highest. And so this is a pretty quantitative way of measuring the expression of the different subgenome messenger RNAs, which, as I mentioned earlier, is just another recombination event. But we can get a bit more granular. So we have multiple different variants in our pool. The vast majority of them are this B1.2 variant, and we can plot the frequency of spike in the B1.2. But then when we compare the frequency of spike subgenome messenger RNAs in both alpha and in delta, we see that recombination event is really suppressed in both alpha and delta. So the spike protein is the frequency of the expression of that spike protein of the subgenome messenger RNA is really suppressed in both alpha and delta. And so we don't know what the mechanism of that is yet. It could be to do with, well, it could be, we just don't know. So we can look into our recombination events and come back to the question of, well, where are they occurring? Is there any nucleotide enrichment? Is there any bias as to where recombination events are occurring? And we know they're occurring at the 3' end. But again, in our clinical samples, what we observed was this enrichment for U's upstream and downstream of these recombination sites. So just like we saw in our clinical samples, we saw this strong enrichment for recombination events just downstream of U-rich regions. And this was true in the B1.2 variants, as well as the alpha variants, we see these spikes. And then of course, as well in the gamma variants, we see these spikes of recombination just after U-rich tracks. So this is telling us that there really is some sort of selection for use for recombination events. Now, when we look at the different variants of concern and that have arisen in the last year or so, there's a little bit more information that we can glean from these variants. So if we look at delta, delta has 2 characteristic deletions in them. Alpha has 3 characteristic deletions. If we look at the nucleotide frequencies surrounding those deletions that characterize each of those variants, which are depicted here, we'll also find those are also flanked by lots of U's, or depicted as T's here. So this ORF1A deletion has this U-rich region. These spike deletions have U-rich regions. There are some other common deletion events we've observed during sequencing, which all have U-rich regions. That breakthrough case from a vaccinated patient that removed part of ORF7A, again, that was also flanked by U's. So it really appears that U's, these frequencies of U-rich regions, are deriving the rate and the positions of recombination events in these novel variants. Now, as you remember, one of the things I pointed out at the beginning is how in the SARS-2 pandemic, one of the signature mutations for that was this insertion in the spike protein around the furin cleavage site, this PRRA insertion. If you look a bit more carefully at this insertion, here's the sequence of the wild-type virus. So this insertion, this PRRA, again, is flanked by U-rich regions. Furthermore, when you passage the virus in cell culture, it's very commonly observed that the viruses kick out those insertions. So if you passage SARS-2 on virus cells, we frequently kick out this QT-QTN region, as well as this PRRA deletion. And so by deleting those regions, these viruses are actually more fit in virus cells. And again, they're flanked by these U-rich regions and are spontaneous. So what we did in collaboration with Vineet Menachery here is we took, using his recombinant system for generating infectious clones of SARS-2 coronavirus, we essentially mutated each of those U-rich regions to remove the U's, and we put C's in instead of U's, or G's, in order to remove those U-rich regions. And then we rescued the virus on cell culture, and we asked a simple question: do we still see the spontaneous deletions arise in cell culture? And so we're looking either for this QT-QTN deletion, which spontaneity arises, or this PRRA deletion, which spontaneity arises. And what we noticed was really kind of unexpected. So when we have both of these regions deleted, both of these regions mutated, so we mutate these U-rich regions, we actually see a completely novel deletion arising during the expression of this mutant virus. And we can quantify that. And in these viruses that had both of these regions mutated, around 30% of the progeny actually had this novel deletion in them. In other words, by tweaking these U-rich tracks, we change the frequency of recombination at those sites telling us quite clearly that those U-rich tracks are deterministic of the rates of RNA recombination and the types of RNA recombination that can occur at those sites. So to wrap up, there's a lot of ongoing questions. So we've clearly observed through sequencing that the rate of RNA recombination changes in different viruses, but that in SARS-2 coronaviruses is strong enrichment for recombination at U-rich sites. And so the open question is, well, why is that? We're just not sure at the moment. And it could be due to the virus itself. There might be RNA secondary structures that favor recombination at U-rich tracks, or there could be some host factors like the APOBEC deamination enzymes, which might be generating U-rich tracks and favoring U-rich recombination at those sites. SARS-2 coronavirus also has this endo-U, this endonuclease that cleaves at U-rich tracks, which is very curious. And so that might be a reason why we're seeing enrichment for recombination at U's, as well as some host factors like RNase L, which are known to cleave at U's. And so knowing that recombination might be, the frequency of recombination might be determined by these host factors, maybe the virus factors, we can then ask, well, do these factors change the outcome of infections? So by changing the rates of recombination, are we changing pathogenesis? And so we can measure the rates of recombination, and we can look in our clinical samples, and we can look at things like these defective RNAs, and we have the clinical data, so we can start to ask questions like, do the frequency of recombination and do the abundance of these defective RNAs actually correlate with specific outcomes of disease in coronavirus patients, just like has been observed in other respiratory pathogens like flu and respiratory syncytial virus. And then finally, TileClickSeq provides a very nice platform for next generation sequencing. So at the moment, we're sequencing for coronavirus, but one of the advantages of our method is we can essentially include any primer that we like in our reverse transcription reaction. And so we don't just have to target coronavirus. And Tori Morris in the lab has demonstrated you actually target multiple viruses at once in the same sample and sequence them all together. And so we designed sets of primers that target both coronaviruses as well as common cold coronaviruses, and then rhinoviruses. And we're developing panels to target RSV and flu, so that in one sequencing reaction, we could sequence the whole genome of any RNA virus, respiratory virus, if it's present. And so that's ongoing in the lab. And I think with the increase in the number of vaccinated patients and people testing positive in spite or showing symptoms in spite of vaccination, I think these co-infections are going to be actually quite common. I think we're going to want to know what is the frequency of other respiratory pathogens that are circulating in our post-coronavirus world. And that's all ongoing at the moment. And so with that, I'll wrap up. And there's way too many people to acknowledge, but I really want to thank everyone from my lab, everyone from the Menacheri lab, and everyone at UTMB for all of their support with what's been a lot of work over the last couple of years. And happy to take any of your questions. And I can see there's a few in the chats already.
All right, thank you, Dr. Routh, for this wonderful talk. So again, you guys can put your questions either in the chat or you can unmute yourself and ask them.
There's a few in the chat already. Unless anybody wants to ask, I can just jump right in. So right at the beginning, someone asks, correct me if I'm wrong here, but don't many viruses have low frequency mutant genomes that are created that are not typically referred to as defective? I think they're called quasi-species, where they would maintain 1 to 3% or something of the population. In this case, how we want to differentiate between defective and so-called quasi-species, or things that actually are maintained in units. Yeah. So actually, I would consider defective RNAs as part of the quasi-species, or really, more technically speaking, the intra-host diversity. The difference here is that they're spontaneously generated all the time. So I think every passage you do, you generate a new pool of defective RNAs. And we don't know what frequencies they're in. That frequency might be different for different viruses. When we passage in cell culture, there might be some defective RNAs that actually outcompete, replicate really well. And we've shown with experimental evolution, we can actually see an increase in the population of those over time. So then I don't just think they're part of the quasi-species. I think they're part of the normal life cycle of any virus, any RNA virus. Any thoughts on how detection of this might or might not be affected by the fact that everyone basically uses the Arctic protocol? Also, how again does your pipeline differ from standard maps that use indels? I think I might have addressed the Arctic protocol questions already. How does the pipeline differ from standard? So Virema is our mapping protocol for sequencing reads. So the purpose of Virema is that it's agnostic. So it'll split the read up into all the bits that don't map and try and find them regardless. So that's kind of unique amongst other mappers. Other mappers will usually look for deletions upstream or downstream. So if you can think of like Bowtie2 or the star aligner, they're usually looking for short, sometimes insertions, but usually deletions. And what I found is that they'll miss a lot of the unexpected recombination events, and they just can't find intergene recombination events. They won't look for a recombination between, for example, coronavirus and the host without some special protocol. So there are tools to do each of these things. There are tools to look at host fusions or chimeras. There are tools to look at splicing or tools to look at insertions. But Virema sort of combines all of them into one pot. It's probably less sensitive than some of those specific tools, but it's more flexible. And because we want to be able to sequence anything that's there, we don't know what to expect. I think that's probably where its strength lies in sort of more agnostic approach. Another question, I've talked to folks who say recombination of viruses is very rare, so it can be dismissed, like an influenza, yet it seems from your talk it could be semi-common. Do you think it is dismissed because do you think it is dismissed maybe because it is dismissed that it ever goes to fixation? We don't actually observe it. Well, hopefully through the talk I've shown that it's really not rare, and that, yeah, recombination events do get fixed. We can see in the variants, the SARS-CoV-2 variants, we see those deletions, they do get fixed. So we know that there are insertions and deletions in the variants of concern that we've got passaging at the moment. Phylogenetic analyses suggest that the actual virus was a recombinant between two different strains. That might have been a very rare event that might have given rise to some which happened to be very fit and then get spread very well. Nonetheless, it's telling us that recombination gave rise to something, even if it's rare. So there's both rare events and there's both common events. Either way, it's giving rise to the variants that we're seeing at the moment. And what we're measuring in patients is with the defective RNAs. Actually, the defective RNAs are pretty common in patients as well. Okay, next question. Is it fair to say ClickSeq is effectively like a probe capture, but still using PCR? And how would it compare in terms of cost, time, potential versus probe capture, like some of the viral probe capture tools out there? It's similar to probe capture in the sense that you have one probe that binds, and then you have one probe that binds. With probe capture, you pull down somehow. Use that probe to enrich for your target. Here, we're enriching for our target by sequencing it and then taking the DNA and then sequencing the DNA. So it's similar in that sense. The advantage of probe capture is you get everything that's covalently attached. So if you have a probe that anneals to, say, spike protein, as long as your genome is intact, you will capture that entire genome and sequence the entire thing if you were to then do random prime sequencing. With TileClickSeq, you sequence anything that's upstream of your probe. And we have thousands of, well, hundreds of probes simultaneously, potentially thousands of probes simultaneously. Okay, next question from Jiang Li says, do the detected recombinations cause change of variant detection from alpha and delta? I'm not sure. What I remember early on with the alpha variants, that one of the primers, there was a frequent dropout of one of those primers because of the deletion. And so there's, I think typically in diagnostics, there are 3 amplicons that are done for PCR diagnostics. And in the UK, the alpha variant, one of them was being missed, quite common, because of a deletion, is what I remember. With TileClickSeq, we have so many primers and they're so close to one another. If one does get lost, it'll be compensated for by other primers that are just upstream. I mean, that's also true of the Arctic, but because we just have so many more primers, we're sort of more robust against that possibility, I think. And should ask, do recombinant and non-recombinant viruses coexist in patients and so will produce viral quasi-species in a patient? Yes, I think so. That's sort of what we're seeing with some of these samples. We see both full length as well as defective genomes. I think the question will come down to the single cell level. If we do single cell sequencing, will we find cells that are enriched in defective genomes and cells that have none? Or will it always be, say, every cell has 2% defective, but every cell has 5% defective? Will it be, if they're generated stochastically and randomly in each round of infection, you might expect that every cell would have a certain dose of defective RNAs. Or it could be determined by what that patient happened to receive. And if they happen to receive a group of viruses from a previous transmitted patient that happened to have lots of defective RNAs, maybe they then will have more defective RNAs. It's a strong bottleneck in the infection, so I don't suspect that's the case, but the inoculum you get could determine the frequency of defective RNAs that the patient has. That's everything from the chat. Is there anyone else like? If you have any more questions, you're more than welcome to email me or tweet me. I think I've got my handle. My handle's just Andrew Routh. You can message me. I'll be happy to answer any of your questions. Oh, there's one more. Sorry. Is the close highly multiplex where you put everything in one reaction? Also, thoughts on semantics of some of the things like recombination versus insertions, deletions, which I feel like most folks say. What you say makes perfect sense, though. Is it fair to say that I feel most folks save the term recombination for one you show between like two infections? Yeah, the terminology is a little bit problematic. I think what's important is the molecular mechanism. If you have a template switching event, you could call that recombination. With small insertions and deletions, it might not be template switching. It might just be maybe the stuttering. Maybe the polymerase just sort of jumps across 3 nucleotides, 6 nucleotides. It might not be the same as a recombination being from one template to a completely other template. So I do think the terminology is mixed up. I think the important thing is molecular mechanisms, but I tend to think in terms of molecules. I think where the terminology also gets a little bit muddy is in terms of defective RNAs, like whether something's defective or whether something's recombinant, or so that deletion in ORF7A might be able to refer to that as a defective RNA. It's defective in the sense that it doesn't generate ORF7A. But it is it's a viable virus. It clearly infects patients perfectly well, causes disease. So maybe it's not defective. So I think the term is sort of relative sometimes. I think it's a gradient of when something's wild type and when something's defective. There are some things that are clearly defective. They're just missing the entire virus genome. They could never replicate. Oh, and is ClickSeq highly multiplex? Yeah, so we can do both. We've demonstrated this arboviruses. So we can target, for example, chikungunya, myaravirus, West Nile, yellow fever, what else? Dengue, all in one pot. And then we've shown that we can find those in experimentally infected mosquitoes without knowing what virus is in there first. So we demonstrated that the highly multiplex approach does work so far. The next goal is to do that with respiratory viruses, like I mentioned. So we're designing rhinovirus primers, flu primers, and so on and so forth.
All right, it looks like that is all of our questions. Thank you again, Dr. Routh, for this really wonderful and relevant talk. And thank you to everybody who attended this seminar, and we'll see you at the next one.
Thank you very much for having me. It's been a pleasure.